DNA replication is where the DNA molecule is copied to produce two identical replicas. Replication begins at a location on the double helix known as origin of replication. Enzymes known as topoisomerase unwind the double helix. Then, helicase starts to split the DNA into two strands by breaking their hydrogen bonds. The single-strand binding proteins are then attached to the unwound template strands to destabilize it. Primase lays down the RNA primers, which provide a starting point of elongation. The leading strand is the 5' 3 strand, which is being elongated continuously. The enzyme DNA polymerase III controls elongation by putting down the complementary nucleotide bases to the parent strands toward the replication fork. The lagging strand runs 3 to 5 and elongates in fragments known as Okazaki fragments. After elongation is complete, two new double helices have replaced the original helix. During termination, the last primer sequence must be removed from the end of the lagging strand. DNA polymerase I then fills in the gaps created by RNA primers and replaces it with the DNA nucleotides, and ligase then comes in and seals the strand.
In the first step, initiation, eukaryotes first have to use general transcription factors or helper proteins to initiate this process. The general transcription factors bind to a promoter called the TATA box, which has many A's and T's that make the strands of DNA to pull apart. The RNA polymerase II can now make a strand of messenger RNA from the template. In the second step, elongation, the RNA polymerase moves along the DNA strand in a 5' 3 direction so that nucleotides can add to the 3 end. For each of the nucleotides in the template strand, the RNA polymerase adds a complementary RNA nucleotide. As the RNA polymerase and the transcription factors move down the strand, the double helix starts to form again. The third and final step is termination, which is the end of transcription. Terminators are sequences that signal that the RNA transcript is complete. Once the RNA polymerase crosses these terminators in the gene, the mRNA is complete and detaches from the DNA.
After transcription comes RNA editing, which is any process other than splicing that results in a change in the sequence of the RNA transcript, such that it differs from the sequence of the DNA template. Pre-mRNA that comes from the transcription changes before it exits the nucleus as mRNA. Pre-mRNA is made of an intron, a non-coding segment, and a pair of exons, the coding segments of RNA. In RNA processing, each end of mRNA is changed. The 5' end gets a 5' cap, which contains a modified form of guanine nucleotide. Then the 3' end is modified with the addition of a poly-A tail. A poly-A tail is added when a sequence called polyadenylation signal shows up in an RNA molecule during transcription. An enzyme chops the RNA in two at that site. Another enzyme adds about 100 to 200 adenine nucleotides to cut the end, forming a poly-A tail. This happens to ensure that RNA transcript is safe from degradation. Then RNA splicing takes place, where a spliceosome, which is made up of small RNAs and proteins, takes out the intron and brings a pair of exons together, creating the mRNA. The mRNA is now ready for translation.
Translation has 3 steps: initiation, elongation, and termination. During initiation, the tRNA attaches to the ribosomal subunit. The tRNA carrying methionine starts in the P site, the middle of the ribosome. Together they bind to the 5' end of the mRNA. They start at the codon AUG. In the next step, elongation, the polypeptide chain gets longer. Another codon is located in the slot next to it called the A site. A new tRNA with an anticodon that matches up perfectly with the exposed codon will land in the A site. Once it lands, a peptide bond will form, connecting one amino acid to that of the tRNA in the A site. After the peptide bond is formed, the mRNA is pulled by one codon through the ribosome, allowing the first tRNA to exit via the E site. A codon is exposed in the A site and the cycle is repeated. Termination occurs when the stop codons UAA, UAG, or UGA in the mRNA enter the A site. Proteins called release factors recognize the stop codon, interfere with the enzyme that forms the peptide bonds. The tRNA separate from the chain and the new protein is released.
Gene expression is when our DNA instructions turn into a functional product. Operons are genes clustered in units that allow the expression of similar genes to be controlled as one. An operon is split into 3 parts: the operator, the promoter, and the genes of operon. The operator is what controls the access of RNA polymerase to the genes. This is located either within the promoter site or between the protein coding genes of the operon. The promoter is where the RNA polymerase attaches. Finally, the genes of the operon is the DNA needed for all the enzymes produced. Once the RNA polymerase binds to the promoter, it transcribes the DNA to RNA. However, there are also regulatory genes that produce inactive repressor proteins, which could potentially block the RNA polymerase from attaching to the operator site of a repressible operon. A molecule known as co-repressor will bind to the repressor protein and activate it, shutting down the operon. There is also an inducible operon, which is typically off, but can be activated by a molecule known as an inducer. The inducer binds to the repressor protein and inactivates it. Once the repressor is inactivated, the operator site is now able to receive the RNA polymerase.